Okay. How are you today? Good, good, good. So my trip to California was excellent in many ways. It was so productive. That was an international, no, actually not international, but it was international in terms of participants, but North American Strawberry Symposium. So a bunch of growers there, and then also scientists working there, California and other states. And I was probably the only controlled environment agriculture person who was presenting hydroponic strawberry production in US there. There are a couple of international people, of course, they are talking about greenhouse and hydroponics, but I was the only one. So I was really sort of happy to be there and learn a lot, from growers and scientists. One of the things I was really interested was photoperiodic response. Right? You learned that last week. Photoperiodic response of strawberry plants. And then I thought, because strawberries are grown in open field, mostly in the US, their issue is limited. Not so much as we have as a greenhouse. We have to determine the flowering initiation and all to target the certain production, time. But for them also, photoperiodic response and how to find out, particularly what they call day neutral varieties, are very, very important. So if you're not familiar with strawberries, okay, most of you are probably not familiar with strawberry cultivars. There are cultivars called day neutral. But that doesn't mean actually day neutral. They call because they see flowers multiple times a year. Okay? So it's potentially quantitative response, short day or long day. Right? But nobody did precise testing over the strawberry plants. Therefore, we don't know exactly what's the conditions that initiate the flowering. So one of the things we are working here is trying to do that kind of, precise tests so that we can find the cultivar-specific requirement of strawberry production. Okay, so relate to that, there are questions, the feedback, seats. There is a good question. So I guess I didn't cover that, but so question was asking tomato, cucumber, pepper, photoperiodic response. What are those? And I can tell what it is for tomato, but I don't know exactly what cucumber and peppers. Peppers probably short day. You can look up the table in the chapter I shared in D2L, and then I think one of the peppers listed as short day. But I'm not 100% sure what is a greenhouse pepper, indeterminate variety of greenhouse peppers. I need to look up. Tomato, it's widely sort of believed as day neutral, indispensable to photoperiods. But there is a couple of research papers telling that it's actually quantitative short day plants. And we really don't know because those research papers haven't done precise experiments, like I mentioned that. Many people mix up photosynthetic lighting and photoperiodic lighting. They haven't separated out. So really need to do in a certain way so that, just like I explained in one slide, you need to separate out the photoperiodic lighting and photosynthetic lighting. So if you are testing photoperiodic response, your experiment needs to be designed so that DLI is equal, across the treatment. So that's very important. But that's a good question though. What is the current understanding of tomato, pepper, all these greenhouse crops regarding photoperiodic response? If you're a graduate student and looking for a project theme, for the review paper, that might be a quite interesting, topic, how they studied the photoperiodic response of those greenhouse vegetable crops and what is the current understanding. Did they appropriately design the experiment to conclude that, or they haven't? Just like strawberry, there might be not so much precise testing going on in that crop. So you know that those plants can flower anytime, right, in the season. So that means it's not going to be qualitative response to short day or long day. We know that already. But is it quantitative response of either way or completely day neutral? So that's something we probably need to find out. Okay. What was that one about? Oh yeah, another good question is I listed in one slide, factors affecting flowering response, and I really didn't mention about nutrient requirement. And there's one big factor, which is nitrogen. The student in the classroom asked this great question. Yeah, nitrogen is one of the factor we know as influencing flowering development. If you provide too much nitrogen, I don't want to generalize, but as far as I know, like tomato does the same thing, strawberry does the same thing. If you provide too much nitrogen in the nutrient solution, they tend to be more vegetative. So that means skipping flowers, so the limiting flower bud development. So it does affect, nitrogen. So it's one of the important things. Any questions regarding photoperiodic response? Please review the recorded lectures if you are not quite sure if you understood quantitative, qualitative response. And then also today is the due date for the review questions. So make sure you understand that. Any other question? Okie dokie, good. All right, so today is going to be CO2. Let me make small. Okay, here. All right, as usual. Okay, CO2. So we basically finished light environment, all right, at the plant level. We come back to the light environment when we go to the canopy, response. But the next topic as a whole plant response, not the canopy response right now, is a CO2. And so there's one reading material written by Hannan, one of the chapters of Greenhouse, the text sort of recommended textbook. So you have that. So it mentions all the way from technology to the sciences in that chapter. So it's a very rich chapter, but I think it's good to read so that you see the sort of whole idea what's going on regarding greenhouse and the CO2. All right, so not just CO2, but I'm going to give the presentation as a gas environment, but focusing on CO2. So what's in the air in terms of gas. So this is a list of component gas composition. So the nitrogen, usually the highest concentration in the air, 79% or so. Okay, and then oxygen 21%, and CO2 is 400 parts per million or micromoles per mole. And then if you express that in percent order so that you can directly compare with nitrogen and oxygen, it's very low, 0.040%. Okay, it's very low, right? But it's increasing, right? We all know that CO2 concentration in atmosphere is increasing bit by bit. I can be a witness of that concentration increase, because when I started my undergraduate degree, at the college, the known concentration in atmosphere was 330. And that was I learned. And then just bit by bit, every time I teach, I had to up the concentration. Now it's 400, right? So I think last year I changed to 380, 390 to 400, because it's reported almost 398 point something. So I round up and then decided to use 400 as the expected atmospheric CO2 concentration. However, depending on where you are located, so the microclimate CO2 concentration is very different. So where you are in a classroom sitting in, you know, people are sort of exhaling, breathing, right? So it's affecting the CO2 concentration in this classroom. In the greenhouse, maybe, you know, again, plants are absorbing CO2. So it's depending on where you are. You know, the city and rural area are probably different also, depending on how much vegetation you are surrounded. Okay, so the unit thing, all right, just a review, percent unit is okay to use, but the scientific literatures, we are supposed to use either micromole per mole or I guess Pascal is used because it's a partial pressure. Okay, the gas concentration can be used, can be expressed using partial pressure. So the total pressure and partial pressure ratio is basically the percent.
For example, if you're doing something specifically the homework, when you're discussing ratio and you were to express the homework as in a ratio of percentage, is it like, can we just assume that, or should, when we're writing out assignments, like something that's not as particular as literature, should we actually like write that out, or is it assumed in something that's more casual like that?
You're talking about gas concentration?
Just ratio.
Yeah, so the percent ratio is better to be avoided as much as possible, unless it is necessary. Because some people actually specify what the ratio is. For example, percent in the parentheses V/V, that means volume per volume ratio. So that is considered as acceptable. However, it's not the ideal. So anyway, yeah, so that's why we encourage that micromole per mole in place of parts per million or percent, okay? And then sometimes you see sort of strange unit like mole per cubic meter or grams per cubic meter for CO2 concentration. I have that example in the exercise today, but it's mostly for, I have seen as in a modeling simulation type of paper, because it's easier to compute that way. Otherwise, you have to convert based on the temperature. So it's not the usual situation, but it is also used in literatures for specific area. Okay, so, all right, so CO2, as you know, affect photosynthesis, and you may or you may not know CO2 concentration also affects stomatal opening, particularly if it is high CO2 concentration. Plant leaves are subject to high CO2 concentration, and then the stomata tends to reduce the opening a little bit, sort of adjusting. So that's a well-known response of the plants to high CO2 concentration. So we say, you know, the conductance changes under, you know, high concentration of CO2, and that, you know, eventually affect the photosynthesis and transpiration rate. So you see that in the exercise today. So oxygen concentration also affect plants, and that is through the respiration. Dark respiration and photorespiration both are affected by oxygen. So oxygen concentration is a percent order, 21%. And then in that case, you know, much higher level than CO2. So to affect the respiration, oxygen concentration needs to be reduced by maybe a half, 10% level or 5% level, slight change doesn't affect the respiration rate. Okay, so it has to be large change, enough to influence the photosynthetic rate, by reducing respiration. Okay, so if you're not remembering well, net photosynthetic rate is the real or gross photosynthesis minus respiration. So if respiration rate is reduced, that means net photosynthetic rate is increased, even though gross photosynthesis is not changed. Okay? All right, so that's why arrows goes this way, respiration and then net photosynthetic rate. Okay? All right. Ethylene, this is one of the hard words to pronounce for me, but ethylene is basically the gaseous growth substance to give plants influence. And then we actually talk about ethylene when we talk about post-harvest environment, because post-harvest ethylene is strategically used to accelerate the ripening or strategically removed to increase the shelf life. Okay, so that's ethylene, and it exists in the air because plants basically generate the ethylene gas from the tissue. Particularly in a closed environment, it accumulates. In the open air, probably you can't detect ethylene in the open air atmosphere, but in a closed environment, you would. However, the concentration range is very low, parts per billion. So 10,000, less, 10,000 times less concentration than parts per million. Yes.
Is the ethylene given off when plants ripen, or is it used as a trigger for
actually, both ways.
Okay.
Giving off, and then also the accumulated ethylene would give the acceleration. So it's a sort of.
When you talk about bananas, like if
Yeah. If you have a sort of unripe tomato, put next to banana, then it accelerates. So that's we talk more about that in post-harvest, which is probably sometime toward the end of this lecture. All right. Okay. So the CO2 is very important, right? Photosynthesis. CO2 is the carbon source for the plant, and if you look at the elemental concentration making up the dry mass of the plants, plants 90% or more sometimes water, right? And then if you look at just the dry mass, okay, after removing all the water, and then the 45 to 50% is carbon. So the CO2 is the carbon source of building the plant, body, dry mass. And plants fix CO2 by photosynthesis, taking up CO2 and then releasing oxygen, so the exchanging. So the because CO2 concentration is so low, it influences a lot and then fluctuate a lot. And oxygen concentration, because the background oxygen concentration is so high, it's a percent order, therefore the small change in oxygen concentration by photosynthesis doesn't seem to influence a lot in the environment or to the plants. Okay? All right. Okay, so the CO2 response of typical plant photosynthesis. So there are the C3 response and C4, and C4 is relatively insensitive to the CO2 concentration. But our study is focusing on C3, which is a greenhouse crop. Okay, so if you see that, so that this one is very similar to light response, right, saturation response. Now we have the CO2 concentration in x-axis, and then net photosynthetic rate as y-axis. This is actual measurement data provided by Ryo Matsuda. He's a former member of my lab. Measured I try to remember what was the crop, either spinach or tomato plant photosynthetic rate. So So something is limiting, therefore it saturates, right? Okay, so what would be the something, typically? The CO2? What's that? Light, yes. So the light is the limitation, therefore CO2 response saturate. So unless you increase the light intensity further, it doesn't increase. The increasing of CO2 doesn't increase. But look at the response. It's about 1,000 or 700-ish in this case to saturate, and it's close to the compensation here. He hasn't quantified the compensation point, but it's probably less than 100 micromole per mole, right? So the same characteristic values you can get from CO2 response, just like for the light response of photosynthesis, saturation point or saturation concentration, which is generally 1,000-ish to all the way to 3,000 micromoles per mole, and then CO2 compensation point 50 to 100 micromoles per mole. So the CO2 compensation point again is the CO2 concentration that balances the gross photosynthesis and dark respiration. Well, actually, all the respiration rate, not just dark, but photo respiration included. Yes.
I guess it's a two-part question.
Mm-hmm.
You can increase the CO2 concentration up to 3,000 micromole per mole.
So technically, can we increase the CO2 up to 3,000? Is that your question?
I guess the slide is saying that you can.
Yeah. Oh, yeah. Technically, yes. You can increase any CO2 concentration as long as you have a gas.
CO2 levels and yield?
That's going to come up soon. Yeah. So his question was the influence to the yield, influence on the yield. So, okay. Good question, TJ. All right. Yeah, so that's the typical response. So, so the light and CO2 are two major limiting factors affecting photosynthesis, right? And then uniqueness of a greenhouse is you can actually inject CO2. You can actually inject CO2 and then create the high CO2 condition if you want to. Okay, so that's why CO2 is strategically used in greenhouse as a sort of alternative to increase the light. So I'm going to explain using a few slides what it means. Do you have a question?
So sometimes in open field it's good to have some source of CO2, like for example some pollution source, I don't know, burning some kind of thing, whatever, that produces CO2 in order to increase the photosynthesis in an open field. This is an hypothetical case.
Okay, so the question is, I couldn't quite follow the procedure, but the open field, it is generally very challenging to increase CO2 concentration.
For example, in Mexico, there's usually, since it's a rural area, lots of people burn trash, whatever.
Yeah.
So that increases the levels of
CO2, yeah.
So
Yeah.
is that a good thing?
Okay, so the question is, for example, depending on rural areas and then people burning trash around the field, is that a good thing or a bad thing? Well, in terms of CO2, it's probably adding CO2 to the air, so that is probably affecting, but you need to burn a lot of stuff. And then that might generate other gases that are toxic. So it's not, 100% good. Good example I have, that relate to your excellent question. So in city-based urban farming, and I know this greenhouse on top of the roof in New York City, they don't have to do CO2 enrichment during winter time, even though, the greenhouse is closed. And then they are thinking because outside level is so high, people or cars and activities, so the general level is higher, therefore they don't need as they normally do in rural area. So so the background CO2 concentration is probably important in open field conditions, yet altering that concentration, increasing particularly, is challenging. It's not easy. So there is a research, as a what was the terminology? I try to remember. So that it's an open air CO2 field atmosphere CO2 enrichment experiment to mimic the high CO2 concentration, the global warming and high CO2. But to do that, the design of the field is very sophisticated. They have many injection pads surrounding the field, and then they inject the CO2 depending on the wind direction and wind speed, so that particular field plot is under high CO2. So it's not easy to do open field CO2 enrichment because you lose. The air is moving. So it's not easy. But good question. All right, so quickly, so if you see the light response curve under low CO2 concentration and high CO2 concentration, it basically increase, right, the photosynthetic response by increasing CO2. All right, and then, so at given light intensity radiance, photosynthetic photon flux, you probably see the increase by increasing CO2 concentration. And that's why I'm talking about the increasing CO2 can be supplemental to the light intensity. So the increasing light intensity in a greenhouse or in any controlled environment facilities, the only way to do that is putting artificial electric lightings. And that is probably more expensive, but yet if you can increase the CO2, you can basically achieve the same influence, whether increasing light intensity by putting a lot of money or minimum amount of investment to increase the photosynthesis by CO2. Okay, so that's why CO2 is sort of practiced in particularly winter time when DLI is limited. Okay, when DLI is limited, or early in the morning when light intensity is still low, but you want to maximize the production by increasing CO2. Okay, and then we talk more about CO2 enrichment practice when we talk about greenhouse system, because depending on the ventilation rate, it is very challenging to increase the CO2 in the greenhouse also. Okay, so the CO2 enrichment in greenhouse, just introduction for the future lecture I will have toward the end. Two different kinds of CO2 generally is used. One is liquid CO2, so the pressurized liquid CO2 in a tank, which is a pure CO2. And the other one is a combustion system, usually natural gas, and it has to be very pure. So usually high temperature combustion system. But either way, this is a picture in California. It's a large greenhouse industry. So those are the big tanks containing CO2. So if growers have access to relatively low price CO2 gas, then this is a great way to do the CO2 enrichment or the combustion system. All right. Okay, so this is our data. I believe this is a CO2 response and light response sort of combined for tomatoes. So this is an instantaneous response. Question? Are you okay?
I was just wondering how they made it. The liquefied CO2, they just burn natural gas and then pressurize it?
That's a good question. I don't have the answer. Yeah.
It seems like
There's a processing factory just to reduce, produce concentrated gas.
Because it seems like if they're just burning natural gas anyways, then it's a huge waste of
Yeah.
energy, because then they also have to expend a lot of extra energy to pressurize it.
Yeah. I don't, the question is about how to make the gas tank for the CO2. I will look up, and you can look up too, and find out. Do you know?
It involves just they really compress just air.
Oh, really?
And fractional distillation. I think that's how they produce nitrogen gas as well. I might be wrong, like argon. But yeah, you just compress the air until you have the different gases forming liquids at different temperatures relative to
Ah.
their boiling point.
But the CO2 is such a low concentration in the air.
Yeah.
Yeah, so
Like doing that with just atmosphere.
combination maybe. Combination maybe.
Yeah, some other form
Yeah.
of concentrate.
Yeah. But anyway, good question, you guys. Right. So this slide is showing the instantaneous response. Okay. So let's orient the figure in here. So the light intensity, photosynthetic photon flux, and then net photosynthetic rate. And those are the photosynthetic response to the light intensity at different CO2 concentrations. So this is atmospheric CO2, and double that, and then about double, boom, 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 increase like this, right? So it's amazing. However, this is instantaneous response. So the plants have sort of unfriendly to growers, mechanism. They get adjusted. If we increase the CO2, they try to reduce the stomatal opening and then reduce the impact of CO2 concentration. So actual impact is not as great as you can see in the instantaneous response. This is the actual response in terms of yield. TJ asked. So long-term influence, because of the acclimation of the plants to high CO2 concentration, is much, much smaller. So this is a sort of reported increase of yield or growth to the high CO2. And then typically the CO2 enrichment research is done around 1,000 micromoles per mole. So it's about the saturation point of the CO2. And then, excuse me, I think the class is going on. Oh, sure. So, okay, so if you look at the table, I have two columns. One is mature plant, and the other one is immature crop. That means young seedlings, vegetative, young seedlings. So if you look at that, the response reported is much higher for vegetative plants or the young seedlings compared to the response for the mature plants regarding yield. So the tomato, for example, is only up to 20% increase by CO2 enrichment, around 1,000 micromoles. And yeah, so I probably missed the explanation. So this N number is number of papers, okay, reported papers. So this is just summarizing how many papers reporting for rows, the final yield, and tomato, and young seedlings, and lettuce. So you see that lettuce is also typical young seedlings, which relatively short period of time exposed to the high CO2 have much higher response compared to the prolonged, long-term response of the mature crop. Okay? So that's sort of expected yield increase. All right. So acclimation process to high CO2. One, I mentioned that instantaneous reduce the stomatal conductance. So just adjust, not closing completely, but adjusting. So it's sort of, saving energy or something. Since CO2 is available so much in the air, just reduce the closing or opening. And acclimation to high CO2 is a long-term adaptation to high CO2 environment. So that's why a lot of studies done in acclimation process. And downregulation of photosynthesis, which is known as part of acclimation process. So the CO2 is available and high, increase of photosynthesis, and that accumulates sugar in the leaf, and then that high sugar concentration in the leaf reduce the enzyme involved in capturing CO2. And we talk about this when we talk about sink-source relationship, okay? So don't worry too much about this mechanism at this point. That's related to Rubisco. Okay, so more important thing today is a different look you want to do, the CO2 influence on the photosynthesis of the leaf. instead of biochemical reaction pathways of photosynthesis, or instead of enzyme reactions of photosynthesis, we want to see the influence of CO2 through the diffusion of the CO2 to the stomata, through the stomata and through to the chloroplasts. Okay, all right. So the entrance of CO2 gas, stomates or stomata, right? Okay, and then the site of photosynthesis is chloroplast. All right, you know that plant biology or biology 101. And so here is my simple cross-sectional stomate. Okay, all right, and then so CO2 in the air, and CO2 actually reach the chloroplast. So certain concentration of CO2 exists in the air, and then CO2 at certain concentration in chloroplast. Okay, maybe dissolved in a liquid in a chloroplast or intercellular space. All right. Okay, so we can look at this as a diffusion. Okay, because plants are not absorbing, right, actively absorbing CO2. Not inhaling CO2, right? They are not doing that. They don't have any pumping system or filtration system. They are just passively accepting CO2. And what is the mechanism of the passive movement of CO2, which is diffusion? Diffusion. What is diffusion? Diffusion is the like CO2 gas molecule traveling from high concentration side to low concentration side. Okay, high concentration to low concentration. So if the concentration difference between those two sites, we are just simplifying the system. So we are looking at CO2 concentration in the air and photosynthetic site, which is chloroplast. If you look at just those two concentrations, if the concentration difference is greater, then you would expect higher diffusion going on, right? Because the driving force of diffusion is the concentration gradient, right? All right. Okay. All right. So any questions so far? Caitlin.
So when you're putting, when you're adding CO2 to an environment, specifically in hydroponics, do you typically measure, or is the dissolving of CO2 into water a concern? Or like?
In the water?
Yeah,
In a nutrient solution?
correct.
No, we don't, because the leaves are the concern of the photosynthetic activities. Unless leaves are, you are growing aquatic plants.
Yeah, I just wasn't sure if it affected like the nutrient solution, like if there was an interaction between the nutrient solution and the added CO2. to
The pH probably would be changed if you bubble the CO2 in a nutrient solution, but usually we are talking about parts per million or micromole per mole order, so not so much, I would say.
It seems in the desert it wouldn't be as good of an idea to do the CO2 enrichment, especially during the summer because of the amount of ventilation required to cool the greenhouse. So you're evacuating all of the CO2 you're producing before the plants do.
Yeah, so TJ is asking the efficacy of CO2 enrichment during summertime when greenhouse is venting a lot. And you are 100% right. It's very challenging to do CO2 enrichment during summertime in a greenhouse. We get back to that point when we talk about greenhouse system, okay? All right, so any questions regarding diffusion? All right, so I'm going to introduce some of the equations here, okay? But it's very simple equation. Right, so diffusion, all right? it's the same thing, all right? So the different orientation. My favorite simplified stomata here, opening, right? And then the CO2 concentration in the air and CO2 concentration in the chloroplast, and that is the driving force. Greater the difference is, then more, driving force of photosynthesis. So the photosynthetic diffusion is the flux of photosynthesis. All right, so there are actually resistances in the path of CO2 diffusion, okay? When stomata close, then resistance increase or decrease? Stomata close, resistance increase, right? When stomata is fully open, that's the minimum stomatal resistance. So that's R sub S written in here, okay? And then above that, there's another resistance we call boundary layer resistance, and then that's something you learn in one of the future lectures when we talk about wind. But there's a sort of resistance created by the air movement around the stomata. So if the air movement is limited, there's a lot bigger resistance involved in there. So that's something you learn. So just don't worry too much about today. Just remember there's another resistance above the stomata resistance. Inside there is what we call mesophyll resistance, because the CO2 gas needs to penetrate through the tissue and reach the chloroplast. Therefore, there's another sort of generalized resistance in there. And then the connection of those three resistance is basically series, not parallel, okay, in this simplified model. So the boundary layer resistance, stomatal resistance, and mesophyll resistance. Okay, so to express the relationship, we are going to use analogy of electric circuit. Okay, so the driving force is a concentration difference between CO2 concentration in air and CO2 concentration in the chloroplast. That's the driving force, which is very similar to the electric potential. Okay, the difference between the electric potential from point A and point B. I think I need to do Elmo here so that you guys can see. Okay, so in an electric circuit, here is one circuit, right? And then you measure the voltage, right? And then this is electric current, which is I, right? And then resistance is the value of R, okay? And then what's the relationship between those three in an electric circuit? The voltage is the electric potential, or difference between the electric potential of point A and point B. So it's a basically driving force of moving electrons in the system. And then there's a resistance, right? Electric circuit. And then the current. Okay, so what is that? So for example, I equal, using V and R. Anyone? Yeah. V over R. And what do you call this law? Ohm's law. Yeah. Bingo. Okay, we're going to use Ohm's law for explaining photosynthesis. All right? So in our photosynthetic system, right? So it's a simple stomata. And then let's just focus just one point, stomatal resistance, all right? And then this is the CO2 concentration in the air, CO2 concentration in the stomatal cavity, all right? And then photosynthetic rate is P, okay? And then resistance is stomatal resistance RS. So how do you express this relationship? By the way, the driving force, similar to the V, is actually CA minus CC. So if this is the case, electric potential B1, B2, it's probably not a good way to express that, but the concept is basically that. All right? So the difference in terms of electric potential divided by resistance is the flow. Okay? So use that. Okay? How do you express this? P, our P equal the driving force, Ca minus Cc, right? The difference of CO2 of the diffusion divided by Rs, resistance. Okay, good. Are you with me? It's very important. Now it's so much easier to understand, the influence of CO2. So if you increase the CO2 concentration in the air without changing anything, so that means this term is going to be greater. So that's CO2 enrichment, right? So that means Ca minus Cc, the value is going to be greater, right? So that means even without changing R sub S, the P is going to be greater. Make sense? Opposite case. You're not changing CO2. You're not changing internal CO2. It's uncontrollable for you. You are giving a great environment so that plants are not under water stress. Okay? So the resistance is minimum. Resistance is minimum. Now something happened to your irrigation system and then didn't irrigate enough. And then plants are under stress. So as soon as plants sense the stress, they start closing stomata. All right? And that means resistance increase or decrease? Increase. So denominator is increasing now. So that means reducing photosynthesis. Super easy, right? Super easy, straightforward. Okay. So this is the equation we just worked out together. This one I have instead of stomatal cavity CO2 concentration, I have still CO2 in a chloroplast. In that case, I have to include mesophyll resistance. But the R is a total resistance, so the all resistance, so the big resistance in here. Okay, if you need to express the total resistance using individual resistances, then you have to sum up. Okay, because it's a series connection. The total resistance is the sum of the individual resistances. You probably learned that in physics, I think, introductory physics. So R sub A, R sub S, R sub M, summation is in the denominator. Okay, good stuff, right? Okay, now if you look at the photosynthetic rate response relative to the total resistance, R equal R sub A, R sub S, R sub M, it's going to be this inverse response, right? So the reducing photosynthetic rate by increasing resistance, like that. When the Ca minus Cc is not changed, constant, okay? So resistance seems to be very, very important, right, in the photosynthesis. Okay, since chloroplast CO2 concentration is very difficult to actually measure, there are research groups actually measuring CO2 or estimating CO2 concentration in the chloroplast and try to reduce the resistance in the mesophyll by molecular approach. But you don't need to worry about this time. So I want to sort of simplify the system by eliminating mesophyll resistance. And then we just look at this, CO2 concentration in the air, CO2 concentration in a stomatal cavity, or we call intercellular CO2 concentration. And then now we have reduced the number of resistance from 3 to 2. Okay, one is a boundary layer resistance in the air. The other one is stomatal conductance. And this intercellular CO2 concentration is not constant value. It's changed because the CO2 is supplied from the air, right, according to the resistances. and then also consumed by photosynthesis. So the CO2 concentration in the stomatal cavity is the result of two, supply and demand. Okay, and this is also very important to understand the plant physiology. What is the intercellular CO2 concentration? And there is a way to understand or estimate what is the C sub I, intercellular CO2. So instead of using CO2 concentration in the air to explain the plant response to CO2, particularly physiologists prefer to use intercellular CO2 because that's more direct influence. You know, the air CO2, even air CO2 is changed, intercellular CO2 may not be changing. And then so effect of intercellular CO2 is more direct than the air CO2. So that's why you may see in the literature talking about intercellular CO2. Not so much in application engineering technology papers, more for photosynthetic or physiological papers. So if you see intercellular CO2, and that's estimated based on estimated resistances and available CO2 concentration. So blah blah blah. Yeah. So this is briefly showing, but it's not so critical. So I'm going to skip this. But basically, if you know the resistances using the measurement of transpiration rate, then you can actually estimate what is the intercellular CO2. So it's not so critical at this point. All right, so factors affecting resistances in CO2 diffusion pathway over leaf. I mentioned that boundary layer resistance, and then we will be learning this a lot in coming lecture. And the value of resistance is 10 to 100 second per meter. Okay, this is the resistance unit, second per meter. So basically how many seconds it takes to travel 1 meter distance. Okay, so the greater the number, then it takes so long. So that means more resistance. Okay, and it's affected by wind velocity and surface viscosity, and that is again topic of future lecture. Stomatal resistance in the range of 77 to 250 reported when open, and that is species, dependent, and then also growing conditions dependent, because plants change the distance. Plants change the density of stomata. How many stomata, stomates per square millimeters, It changes depending on high CO2 concentration tends to reduce number of stomates in the leaf surface. So that is changing depending on the conditions. But anyway, so it's about the same order, right, as boundary layer resistance when it is open. But of course, when it is closed, the value is jumped to much higher, more than 10 times. So 5,000 second per meter, right? By the way, 5,000 seconds is what? More than 1 hour, right? 3,600 is 1 hour. So it takes 1 hour to get to 1 meter. It's really, really slow, right? So you can see how resistant is it. And those, numbers affected. So there is in between, okay? There is in between. So it's fully open, fully closed, and then also in between. A little bit open, very close to wide open, right? And then also again the size of stomata, and then also density of stomata affecting those resistance value, okay? Not just open and close. And plant water status again affects the opening and closing of stomata. When plants are stressed, water stressed, and ABA is produced, and then give the signal and then close the stomata quickly. And then also we touched this upper level plant physiology. Blue light affects the opening and closing of stomata. when blue light hits the stomata, they tend to open, okay, or increase the opening. And then I mentioned that CO2 concentration affects stomata opening too. And mesophyll resistance, this I don't exactly know how they measure this, but there's a way to measure that, and then that's in the order of 100 to 1,000 seconds per meter. So if you look at those numbers, all three resistances are basically quite, important, and you can't ignore, right? All right. And then we talk more about boundary layer resistance, which is a lot of fun, by the way. But anyway, so that's the level of resistance. Okay, so if you are not familiar with the unit, so this is the resistance unit, second per meter. And then conductance and resistance are basically the flip side of the same thing, right? So the resistance is the inverse of conductance. You know that, okay? So the unit of conductance could be meter per second, or you might see cubic meter per square meter per second. If you try to interpret that, so 1 cubic meter of gas transferred per unit area, the section per second. So that's the unit. Or if you can understand this, how much time you need to travel 1 meter, that's the inverse. Actually, if you clean up, the canceling square meter component from cubic meter, then it generates the meter per second too. And then if you want to interpret the meaning of that, is probably how much traveling, distance per second. So that's one way to understand the unit. You also see conductance and resistance in terms of moles per square meter per second, which is the same way how much mass of gas traveling through that section of 1, square meter per second. If it is faster, then you have a massive amount of CO2 going through that section, so that's a conductance. And then if you get the inverse of that, so it's so hard to interpret this unit, but if you understand this and understand the resistance is inverse, then you understand this, because now cubic meter and second is numerator, and then mole is the denominator. Okay? All right. Because you've done SI units, I'm just playing with that. All right. So that unit is very helpful and helps you understand what it is. Okay. Now, how are you doing? Doing okay? It's a little too warm? Not so much? Okay. So you have a question?
I was just curious,
Yeah.
did we discuss what ABA was? I think a few slides back with,
Abscisic acid, ABA, yeah. Yeah, it's a plant growth hormone or growth substance generated by plant, and it is usually involved in many stress-related responses. Sort of signaling compound going through the system of the plant. I think generated in the roots, and then transported to the, I need to look up probably, but was that correct? Recent people who learned? I forgot. But anyway, so it's a signaling compound, ABA. If I'm wrong, I will correct that tomorrow. Wednesday. Okay, any question? All right, good.
When we're talking the seconds per meter,
Yeah.
is that as if we're taking like a column of CO2 and each time it would take?
No, it's a mass, so that the big mass, so that sort of. So that's why, yeah. So this probably much easier for understanding one section of, the cross section of 1 square meter, and mass of gas is passing through, and then. No, when you apply stomata, that's going to be stomata, opening, equivalent to 1 square meter, which is not really, but you sort of extrapolate the size to 1 square meter. So let's assume big stomate having 1 square meter opening, and how much CO2 is moving is the CO2 conductance, cubic meter per square meter per second, right? And then inverse of that is actually second square meter per cubic meter, but if you cancel it out, it reduced to this simple form, second per meter. Okay, the same way, if you cancel those out, meter per second, right? So the same way, second per meter. But if you want to interpret as it is, second per meter, what it means is basically how much, whatever the size, whatever the opening space, canceling out, how much seconds, right, needed to sort of travel 1 meter length.
For the gas.
Yeah. If you look at, if you can look at 1 molecule of CO2, that's probably easier to understand. Okay, good. All right, so I'm using the same analogy of Ohm's gas diffusion to transpiration. Why I'm talking about transpiration when I'm talking about CO2? It's because it's very important to understand two diffusions going on through the same opening. Okay, one is the CO2 diffusion, the other one is water vapor diffusion, right? So the water vapor diffusion, which is transpiration, right? Okay, the plant's losing water, and that usually mainly from stomata, through the stomata, and some from the cuticular layer, epidermal layer, but mostly in the stomata because it's open, and then inside is completely saturated air with water vapor. So the diffusion direction is opposite. Now it's from inside to the outside. Okay, from inside to the outside. So we can consider that stomatal cavity is saturated at nearly 100% humidity, and it comes out, the moisture comes out from the stomata, and mesophyll cells are constantly supplying moisture to the stomatal cavity. That's why it's nearly saturated, right? So using the same understanding now, the difference between the concentration of water in the air in the stomatal cavity and water vapor concentration in the air, moisture concentration, humidity, is the driving force of this diffusion. Greater the difference is, then more transpiration, the loss of water happening, right? So we can actually use the same analogy, stomatal resistance, boundary layer resistance, and the concentration in the leaf or stomata and concentration of the air, and what is the equation, right? Okay, anyone? E, A, E, L. Boundary layer. Okay, so transpiration rate or TR. Anyone?
EI or EL minus EA
Yeah,
over RA plus RB.
that's right. Right, super simple. Okay, you are the expert. But actually, you did an excellent, first step to understand plant growth modeling. Okay, you just modeled the response of photosynthesis and transpiration using the simple equation using the theory of Ohm, right? Ohm's law. So now I'm going into a little bit, flavor of modeling and simulation. I'm not going to talk too much about modeling and simulation in this course, but if you are interested, because what we do in this course is quantitative understanding of plant responses. And often that approach leads you into this modeling and simulation. Maybe one more lecture probably I have talking about modeling, but not too much. I try not to overwhelm you with bunch of equations and stuff like that. But this is just the introduction how interesting the plant modeling approach is. So modeling, you need input and output and define the system, and then basically describe the response of the system, in this case plants, using mathematical equations. Okay, once you express mathematical equation, just like you experienced that using Ohm's law, understanding the influence of CO2 enrichment, understanding influence of resistance increase by water stress, it's so easy because you can see how vigorously it responds or how non-vigorously it responds. Easily you can tell, right? So, so the system you are talking about could be very complex. So in that case, your model description based on theory sometimes really challenging. And then in that case, you just see the response and describe what it looks. For example, photosynthetic response curve, saturating with increasing light intensity is sort of empirical. We know that how it responds. Therefore, we are using that response. So there are two ways: theoretical models and descriptive model or empirical model. So the descriptive or theoretical model, say that again. Theoretical model, one example is just like we did, diffusion, Ohm's law. That's a very theoretical. Okay. So I'm going to show you a few examples of modeling. No need to understand this, okay? No need to memorize the equation. So this is a Campbell 1977, relatively old approach of photosynthetic model. But look at that. They are using Ohm's law for the first step of expressing photosynthetic rate relative to the CO2 concentration, and then also the summation of all the resistances to get the total resistance. Exactly what we did. And then other equation, which is if you took chemical engineering, I think, you learn Michaelis-Menten equation. That's an enzyme reaction because photosynthesis involves enzyme reaction, substrate, and kinetics. So you tend to use this sort of empirical expression of the reaction. No need to understand what it is, but it's coming from the enzyme reaction, okay? And then this one is actually quadratic solution of the equation. But anyway, so combining those actually nicely, you can compute the photosynthetic response using CO2 concentration as a factor, temperature as a factor, and light intensity as a factor, and I think, yeah, so the next one. So I created, just following what they did, what he did, created a spreadsheet, which is also available in the D2L. Showing the photosynthetic response. So those red values you can change. So there are 3 different photosynthetic response curves shown in here. The unit is original unit they use, so watts per square meter. I wish I can change that to micromoles, but I'm using the original, the equations. The CO2 concentration is weird value, grams per cubic meter, but I see this here. I know that 0.7 is about 400 micromoles per mole. So the 3 3 conditions at the same CO2 concentration. And then here, the red one, I have a total resistance, which is a summation of stomatal resistance and boundary layer and what's the other? Mesophil resistance. So right now I'm showing 3 curves having 3 different resistances total. So the 200 relatively nice environment, 400 and 2000 relatively closed. So if you change that, you can see the response. So change to all the same, 200. So the fully open, you know, the stomatal conditions, that's why three, you know, curves overlap each other because all are in the same condition. And I'm going to change the CO2 concentration. Let's see, plants are growing in a contained environment. CO2 is limiting. Maybe the concentration reduced to 0.5, which is probably 300 parts per million or so. Then you can see the response. And then let's see, I'm going to see, you know, very high, let's see, experiment 3, high resistance. Maybe resistance is 10,000. And that reduce the, you know, the photosynthetic response. So it's, you know, again, you kind of see that, the sort of power of the simulation and modeling and simulation. Okay? There are a bunch of different models actually available. The second one is a sort of empirical, so the shape, you know, you have a light intensity and light compensation point and maximum photosynthetic rates as the important characteristic values of photosynthesis. You learned that in my second week of lecture. And now, using those values as a parameters, you can express photosynthetic rate like that. So the Pmax is a maximum, and K is the slope, or relate to the quantum efficiency, and CO2 light compensation point is here. So using this equation, actually apply this equation to the data you get, and then find out what are those parameters, and then actually talk about, you know, for example, salinity effects, which I did in one of the papers on those values, or water stress effect, or cultivar effect, or some other, you know, cool treatments you might want to test in terms of influence on photosynthetic characteristics. So the whole section of this part, you know, the last 5 minutes was how, you know, effective this approach could be. Quantitative understanding, therefore modeling and simulation, testing your hypothesis even before you actually start growing your plants, right? So that's a very important when you do this approach. And then the last one, maybe just for graduate student information, there is a well-known photosynthetic model, which is including biochemical understanding, you know, the process understanding of photosynthesis. And this is a Farker and Sharkey, well understood and utilized. I'm not going to explain too much detail, so this is just to mention. But they use also Michaelis-Menten equation, which is the descriptive model of the photosynthesis. Okay, I'm on time. All right, any questions? All right, then. Thank you.